Hey guys! Today we are going to explore a concept called the Red Queen hypothesis. This is an evolutionary hypothesis that was first introduced by Levin and inspired by Lewis Carroll's book Through the Looking Glass. In this book, the Red Queen says to Alice, "It takes all the running you can do to just stay in the same place." By this, she is alluding to the idea that species have to constantly run or partake in a co-evolutionary arms race just to survive. Let's explore this phenomenon using our model organism, the influenza virus, which belongs to the family Orthomyxoviridae. In doing so, we will demonstrate an example of host pathogen coevolution. Influenza is the virus that causes the flu. You have likely encountered it in some form or the other every year. It is one of the most common infectious diseases. Its symptoms are fatigue, rhinorrhea, myalgia, fever, a cough or sore throat, and headaches. There are 3 different types of influenza that infect humans: type A, type B, and type C. Each type of virus is made up of a different genome. Influenza A and B viruses contain 8 gene segments, while influenza C contains 7 segments. Influenza A viruses are divided based on antigenic properties of their surface glycoproteins, which are 16 hemagglutinin and 9 neuraminidase. These proteins can vary in their structure. For example, you may have heard of H1N1 or H3N2. Well, these are just subtypes of the influenza A virus. Influenza viruses have a gene coding for one of 16 possible HA surface proteins and another coding for one of 9 possible NA surface proteins. Of the 144 total combinations that are possible, only 3 HAs and 2 NAs and only 3 combinations, H1N1, H2N2, and H3N2, have been identified in human adaptor viruses. So why is the flu virus the most common infectious disease out there? Well, viruses are constantly evolving through 2 mechanisms known as antigenic drift and antigenic shift. Antigenic drift is described by small changes that occur in the genes of the virus. These changes happen continuously over time as the virus replicates. Viruses have a tendency to mutate their H and N proteins as they replicate, resulting in daughter viruses that have new H and N proteins. As these small changes accumulate, new strains can start to infect individuals who may have been immune to previous strains. So why is it that your body's immune system can't recognize a newer virus? Our bodies produce a protein called antibodies, which recognize viral particles and attack them. As we are exposed to viruses, our bodies make antibodies for that particular virus. However, as antigenic changes accumulate, the antibodies made for the older virus no longer recognize the new virus, and this leads to you becoming sick once again. This is why we are all likely to get the flu more than once in our lifetime, and this is also why new vaccinations are made yearly for the flu. Other ways viruses can change is through antigenic shift. Unlike antigenic drift, antigenic shift consists of abrupt changes in influenza A virus proteins hemagglutinin and neuraminidase. These changes arise through a phenomenon called viral reassortment. Let's just say a virus is circulating through animal populations such as pigs or chickens, and the virus suddenly changes in a way that allows it to infect humans as well. This happens when one cell, let's say pig cell, gets infected with 2 viruses, one that usually infects humans and one that usually infects pigs. The viral genome is divided into segments of RNA which in the cells can get swapped, resulting in an entirely new virus containing either new H proteins, new N proteins, or both. Once again, a virus with an entirely new genome infects humans and spreads throughout the population, since the population doesn't contain any antibodies against it. This is how major pandemics such as the Spanish flu and H1N1 outbreak in 2009 were caused. How do humans evolve and stay protected against viruses? One of the ways in which our immune systems work to defend us against viruses is through antibody production. Antibodies are proteins whose function is to recognize harmful pathogens and bind to them. Binding to a virus neutralizes it so that it is prevented from further infecting the cell. Next, many antibodies assemble together in order to stick offending viral particles together. This is termed agglutination. Lastly, antibodies alert phagocytes to engulf and get rid of these cells. Our immune system response to viruses consists of both innate and adaptive defenses. Innate responses refer to the more constant day-to-day defense of the immune system, so that we will not focus on that here. Most viral infections can be taken care of by the innate immune system alone. However, when replication outpaces our innate defenses, the adaptive immune system must be called in. An important feature of the adaptive immune system is memory. Continuous infections by the same virus are met immediately with a strong and specific response that then stops the infection. When you become immune to infection by a virus, this is because of your immune memory. For instance, vaccines protect us against infection because of immune memory. The first adaptive response against the virus, called the primary response, takes days to mature. But by the time this response has taken place and stored in your immune memory, the next time you have an infection, a memory response develops and is executed within a few hours. A subset of B and T lymphocytes are responsible for this memory response, and they remain ready to respond quickly and efficiently to any future subsequent encounter with a pathogen. The response by immune memory is termed the secondary response, and it is stronger than the primary response to infection. This is how childhood infections protect you well into adulthood, and immunity conferred by vaccination can last for years until a new viral mutation requires the immune system to develop another response. All year long, 142 national influenza centers in 113 different countries collect data of the flu viruses impacting the world's population, specifically monitoring the strains of viruses that make people sick. This is so that they can prepare a vaccine for the most common strain of the virus, so that the immune systems population-wide will be inoculated against the virus, which then leads to herd immunity. Now, why is this relevant to the Red Queen hypothesis? First, we have demonstrated that when a particularly virulent viral strain infects you, your immune system must try to defend itself. It can do so using pre-existing defenses from the innate immune system or more targeted defenses from the adaptive immune system. The targeted defense from the adaptive immune system can obstruct the virus from further infecting cells, and in doing so, destroy it. Second, the virus will then undergo mutation. Evolutionary pressure works in two ways. First, it works to ensure host survival. Second, it works to allow for viral replication. Viral infections and host responses work in tandem to ensure the continuation of a coevolutionary arms race, which then drives continuous selection for beneficial mutations on the part of host immune proteins and viral cells. Since viruses can evolve faster than their human hosts, our current vertebrate innate immune system works best at defending us against ancient viruses rather than current viral threats. The way natural selection has selected for and against certain proteins in our immune system is helpful in describing why we are immune to certain viruses and then susceptible to others. Thanks for watching! We hope you enjoyed the video and are able to identify and understand the concepts behind the Red Queen hypothesis and the coevolutionary arms race of influenza and the human host response.